So now we're going to talk a little bit about DNA replication. So a eukaryotic cell has linear DNA. So if we draw the DNA, it's double-stranded and the bases are going to line up in the middle. Prokaryotic DNA is circular, but it's still double-stranded and the bases still line up in the middle. DNA replication is known as being semi-conservative. So with semi-conservative replication, when we do make our DNA, we end up with DNA that is half old and half new. In other words, the strands of DNA will separate, and each strand will be used as a template to create a new strand of DNA. So the new strands get built on the inside here, and this is what we end up with. So prokaryotes versus eukaryotes DNA replication. So for prokaryotes, DNA is just hanging out in the cytoplasm. DNA is circular. There's only one origin of replication, so the Okazaki fragments can be a little bit longer, and DNA is wound around histone-like proteins. In eukaryotes, the DNA is in the nucleus. DNA is linear. There are several origins of replication, shorter Okazaki fragments, and the DNA is wound around histones. So when we talk about the steps of DNA replication, remember we're starting with double-stranded DNA. And the first step is to open up the DNA, because we need to be able to get into the strand of DNA. So as we open this up, the enzyme responsible for doing that is known as helicase. And helicase is going to travel in one direction, opening up the strands of the DNA. So step one, helicase separates the strands of DNA. In step two, primase is involved. Primase is an RNA primer laying enzyme. So primase is an enzyme that lays down an RNA primer. So we're going to put an RNA primer here. Now there's a top strand and a bottom strand of the DNA. And we label our ends 5' to 3', and then 5' to 3'. DNA can only be made in the 5' to 3' direction. So when DNA is created, we have to create it in this direction. Now, since the strands are running in opposite direction, and helicase is here, traveling in this direction only, one strand, we can lay down a primer at this end, but the other strand, we have to lay it down at this end. And again, that's just because of the directionality that the DNA can be produced in. So the next step, we have DNA polymerase 3. So as helicase opens up more of the DNA, we have our RNA primer that was laid down by Primase, and we have an RNA primer that was laid down by Primase about here, and then our new DNA is going to be made using DNA polymerase. So helicase again is traveling in this direction. Now DNA polymerase can come in here and add a new base. And DNA polymerase can keep adding DNA in this direction because that's the direction that this strand happens to be produced in. This top strand thus is called the leading strand. Because the leading strand is synthesized in the same direction that helicase travels. But with the bottom strand we have a problem. Because DNA is anti-parallel, these strands are going to be synthesized in opposite directions. So this strand is actually going to be made in this direction. Now we have to lay down a second RNA primer as the next segment of DNA opens up. And then we can make DNA backwards again. This bottom strand is known as the lagging strand. And this strand is synthesized backwards in fragments or in chunks, and those chunks are known as Okazaki fragments. So helicase is going to keep going around and opening up the rest of the DNA, and since we're talking about bacterial DNA or DNA replication, this thing is going to keep going around in an entire circle, loop all the way around until it gets back to the origin of replication. So when DNA has been completely replicated, our leading strand looks like this. But our lagging strand looks a little bit different because this one was made backwards in these fragments. So DNA polymerase 1 is going to try to fix this. What DNA polymerase 1 does is it comes in here and gets rid of the primers and replaces those primers with DNA. So if you recall, what we originally wanted to do is we wanted this picture in the end. We wanted the old DNA on the outside and the new DNA on the inside. And the top strand looks okay, but the bottom strand is still a little bit messed up because there's these gaps that exist between each one of the Okazaki fragments. So the final step involves ligase. So what ligase does is glue together the Okazaki fragments to complete DNA replication. So in the end, we have our semi-conservative replication completed. We have the template strands on the outside, and then we have the new DNA that was synthesized. Now these two chromosomes will separate away from each other, and we do this process of DNA replication in order to go through binary fission for a bacterial cell. So for cell division to happen, we have to have this process happen.